Hello everyone. For this particular session, I will be presenting to you another topic, okay, in relation to our subject, medical pathophysiology. So we are now done with lecture one, introduction to medical pathophysiology. And for lecture two, I will be talking about the different accessory organs of the digestive system. So for this particular discussion, I will be showing to all of you, or I will be discussing to all of you, the different functions of the liver, the pancreas, and the gallbladder. Okay, so here are the following topics to be discussed for this particular discussion. Number one, we will talk about the anatomy of the liver. Then after discussing the anatomy of the liver, we will now be discussing the different functions of the liver, the synthetic function, the detoxification and drug metabolism function, the excretory and the secretory function of the liver, and of course, we will also be talking about the functions of the gallbladder and the pancreas. We will also take a look into what are the laboratory tests, okay, available for the diagnostic process for liver function, for pancreas, and for the gallbladder. So the liver is being classified as the chief metabolic organ. Okay, so when you talk about this particular organ, it is really of prime importance that we have a functional liver. Okay, our liver is healthy because the liver is so important that if the liver becomes non-functional, that will occur within 24 hours due to hypoglycemia or decrease in blood sugar. So just like your brain, your lungs, okay, and especially your heart, liver is considered to be one of the most important vital organ of the human body. Your liver receives 15 ml blood per minute. Okay, so it receives 15 ml blood per minute and it weighs approximately 1.2 to 1.5 kilograms. Okay, so we have two lobes. Okay, so your liver is being divided into two lobes. Of course, we have the right lobe and we have the left lobe. And if you want to compare the size of the lobes, the right lobe is much larger than the left lobe. Okay, so always remember, guys, that the right lobe of the liver is 6 times larger than the left lobe. And inside the liver, we have two type of cells. We have the hepatocytes, okay, and we also have the Kupffer cells. Okay, so when you talk about the hepatocytes, okay, when you talk about the hepatocytes, these perform major functions and associated with the regenerative process of the liver. On the other hand, the Kupffer cells are your macrophages, okay? Our macrophages, which acts as an active phagocyte capable of engulfing bacteria, debris, and toxin. Okay, so since the liver, okay, is being considered as one of the most important organ, we have actually natural ways to protect the functionality of the liver with the help of our Kupffer cells, who is said to be the front line of defense for liver. So if there are any bacteria, any other toxin or debris that will go to the liver, the Kupffer cells are the ones who is responsible for the engulfment of this particular bacteria or debris, okay, in the process, of course, of phagocytosis. The cells are also arranged in the lobule, which is said to be the anatomic unit of the liver. So here's a picture, okay, that will show to you the anatomy of the liver. So we all know, right, that the right lobe of the liver, okay, is much larger compared to the left lobe. Okay, so the right lobe is much larger compared to the left lobe. And another important characteristic of a liver is that the liver is an extremely vascular organ that receives its blood from two sources. Okay, so it's a vascular organ, okay, that receives blood from two sources, and this is what your hepatic artery, okay, your hepatic artery and your hepatic vein. Okay, so your proper hepatic artery and your portal vein. Okay, so 25% of blood supply comes from your hepatic artery. So 25%, okay, of blood supply comes from your hepatic artery, and the remaining 75, okay, 75 percent of your blood supply comes from the portal vein. Okay, so approximately there is about 1,000, okay, 1,500, okay, 1,500 ml, okay, 1,500 ml of blood usually passes through the liver per minute. Okay, so the liver is drained by the collecting system that empties into the hepatic veins and ultimately into the inferior vena cava. So let's now discuss the main functions of the liver. So first, okay, we will be talking about the synthetic function of the liver. So when you talk about a synthetic, okay, function of the liver, it is responsible for the secretion of plasma proteins, carbohydrates, lipids, and clotting factors, okay, which is essential for the activation of platelets and the the formation of clot, okay, to impede blood flow, bleeding, okay, of the vessel. So but the main synthetic function of the liver is the production of albumin. Okay, so the liver is able to produce 12 grams of albumin in a daily basis. Okay, then another one is the metabolism of carbohydrates. Okay, so in your chemistry, I think you're familiar with the process of glycogenesis and glycogenolysis, right? So the liver is capable of doing the process of glycogenesis. So from the word genesis, which means the beginning or the onset, right? So in this particular process, glucose is being converted to glycogen, okay, for storage purposes, okay. And also when you talk about glycogenolysis, so from the word lysis, okay, which means the breakdown, so this is a process in which glycogen is being degraded or to being breakdown to glucose, so that glucose will be converted, okay, to ATP for energy production. So aside from carbohydrate metabolism, the liver is also capable for lipid metabolism, and almost all proteins are being synthesized or are being formed by the liver, except for the immunoglobulins and your adult hemoglobin. So your immunoglobulins are also being referred to as your antibodies, okay? And the cells that are responsible for the creation or the formation of your antibodies are your B lymphocytes. Okay. On the other hand, hemoglobin is being formed or being sensitized in the process of red blood cell metabolism. Okay, so the liver also is responsible for synthesizing acute phase reactants. So what do you mean when you hear the word acute phase reactants? These are markers in the body that usually increases or decreases, especially if the body is experiencing inflammation or malignancy. Okay, so we have two classification of acute phase reactants. We have the positive and the negative acute phase reactants. So when you talk about positive acute phase reactants, this will tell you that during inflammation or during malignancies, this acute phase reactants will increase, okay, will increase, or the numbers, okay, will increase, okay, when you talk about positive acute phase reactants. But when you talk about the negative acute phase reactants, during inflammation or during malignancy, the levels will slow down, or there is decrease in the levels for acute negative acute phase reactant. So one primary example for your positive acute phase reactant is your CRP, or what we called as your C-reactive protein. Okay, and another example for your negative acute phase reactants, which will decrease in times of inflammation or any other malignancy, is your albumin. Okay, your albumin. Okay. Okay, so let's now discuss albumin. So always remember, guys, that your albumin is the most abundant protein in the plasma. It is primarily synthesized by the liver, and this is actually used as an indicator of nutritional status. For example, if you want to know whether this child is experiencing malnutrition, we can actually measure the albumin levels. Aside from that, the main function of albumin also maintains osmotic pressure, okay, within vessels. Okay, so when you talk about osmotic pressure, so this is the pressure that prevents the escape of fluids in the blood vessel. So once that there is decrease or low levels of albumin, this will also lead to the decrease in osmotic pressure, leading to edema. Okay, leading to edema or the escape of tissue fluid around the vessel or the blood vessel. So always remember, guys, albumin is the primary cell or the primary marker for nutritional status, and it is also the protein that is responsible in maintaining osmotic pressure. Aside from that, your albumin is also considered as your negative acute phase reactant. So a while ago, I discussed to all of you that uh, when an acute phase reactant is being classified as negative, the levels will usually decreases, okay, during inflammation or malignancy. Lowest plasma of albumins are also seen in active nephrotic syndrome, which is a type of kidney disease. Okay, so we will now be talking about your immunoglobulins. Okay, so your immunoglobulins again are being referred to as your antibodies. Okay, your antibodies. Okay, which plays a major role in the immune defense system. So always remember, guys, that your immunoglobulins, even though they are considered as proteins, but the main difference, okay, just from the other proteins is that your immunoglobulins are being produced by your B lymphocytes, okay, your B lymphocytes, and not by the liver. Okay, so there are five major classes of your immunoglobulins. Okay, so we have IgG, IgM, IgA, IgE, and IgD. And one way, okay, one way also, or one method to classify immunoglobulins is based on serum protein electrophoresis, which will be discussed in the next video. Okay, so when you talk about electrophoresis, okay, we will actually separate the different types of proteins based on its ability to migrate to a positively charged electrode. Okay, so immunoglobulins can be found, okay, in almost all parts of our body, right? Specifically, the blood plasma. Okay, this can also be found in many other body fluids, such as your tears, your saliva, and your colostrum. So the primary function of your immunoglobulin or your antibody is to bind with the antigen. Okay, to bind with an antigen enough to neutralize bacterial toxins or some viruses. So always remember, guys, that the antibody towards a particular antigen should always be specific, okay, in order to induce or in order to create an immune response. Okay, the secondary function of your immunoglobulins is in the process of opsonization. So opsonization is a process in which an antigen are being coated with antibodies, okay, so that they can be more recognizable by a phagocyte. So another function of your antibody is to coat an antigen so that it can the phagocyte can easily distinguish this particular antigen, and phagocytosis will happen, okay, right, okay, or immediately, okay, ang phagocytosis. So if you add, it's like similar to when you cook, right? So di ba when we are cooking, we add some seasoning, right? We add some seasoning to make our food yummier and nice to eat, right? So that is also the process of opsonization. The antibody will coat a particular antigen, okay, so that it can be easily be engulfed or be eaten by a phagocyte, like your white blood cells. Okay. Another function, of course, is your complement fixation. So complement fixation is a mechanism which leads to the lysis or the destruction of a particular antigen or foreign material. So let's now discuss the immune response. So I just want to share to all of you guys the work of your antibody, okay, in terms of destroying a particular antigen. So the immune response is being divided into into two rather. This is your primary response and of course secondary response. So let's now discuss the primary response. So on your first encounter with the antigen, okay, the first antibody that will rise is in the form of IgM. Okay, so this is the first antibody to rise. Okay, and after IgM came your IgG, which is the second antibody to rise. Now, um, the main characteristic of your IgG is the is able to create or able to develop memory cells. So that is one of the special feature for your IgG, the ability to develop memory cells. Okay. Now, let's take for example the chickenpox virus. So once on the first encounter with the chickenpox virus, there is a greater chance that you might be infected. Why? Because your body or your immune system do not have memory cells. And the first antibody that will actually rise is the IgM, okay, which is not able to produce memory cell. But after the infection, okay, you already develop memory cells. Okay, that is why on your second encounter with the same antigen, okay, just like your chickenpox virus, okay, your IgG levels will increase, okay, will surely increase, okay, will peak, okay, mo peak ang atong IgG levels leading to immunity. against chickenpox. That is why if you get infected of chickenpox in the past, okay, there is no there is no chance that you will be infected again with the virus. Why? Because your immune system already developed memory cells, okay, due to your IgG. Okay, and aside from that, always remember, guys, that IgM is actually used to diagnose recent infection. Okay, so your IgM is used to diagnose recent or present infection. Okay, because again, this is the first antibody to rise in terms of infection. On the other hand, your IgG is actually used to diagnose past infection or immunity against the particular antigen. Okay, so don't forget, ha, the first antibody to rise is your IgM. Okay, but it will not lead to the production of memory cell. Okay, your IgG is the one who has the ability to produce memory cells. That's why on the second encounter with the same virus, there is a greater chance that you will not get infected because your immune system already developed memory cells. Okay, so please go on to the next video for the continuation of the discussion.